What is molecular profiling for astrocytoma diagnosis? Imagine trying to solve a complex puzzle where each piece reveals more about the picture. That's similar to what molecular profiling does for diagnosing astrocytomas, a type of brain tumor that arises from astrocytes, which are star-shaped cells in the brain. Traditional diagnosis involves looking at tumor tissue under a microscope, but molecular profiling adds a new layer by analyzing the genetic and molecular features of the tumor cells. This helps doctors understand exactly what kind of tumor they're dealing with, how aggressive it might be, and what treatments could work best. Astrocytomas are part of a larger group called gliomas, which are tumors originating from glial cells. These tumors can behave very differently depending on their molecular makeup. Some grow slowly and respond well to treatment, while others are more aggressive. That's why molecular profiling is so important. It helps distinguish these differences with more precision than just looking at the tumor's appearance. The process involves testing for specific molecular markers and genetic changes. For example, mutations in the IDH1 and IDH2 genes are common in certain astrocytomas and are linked to a better outlook. Scientists also look for chromosomal changes like the co-deletion of parts of chromosomes 1 and 19 or mutations in the TERT promoter gene. Other genetic alterations, such as changes in BRAF, CDKN2A, or EGFR genes, can also provide clues about how the tumor might behave. To do this testing, labs use techniques like immunohistochemistry, which detects proteins linked to genetic mutations, fluorescence in situ hybridization, which finds chromosomal deletions or amplifications, and DNA sequencing methods, including next-generation sequencing, which can identify tiny genetic changes. Methylation profiling, which examines epigenetic modifications, has become a powerful tool for classifying tumors based on their DNA methylation patterns, often providing more accurate diagnoses than traditional methods. Molecular profiling is not meant to replace traditional pathology but to work alongside it. The World Health Organization now recommends combining molecular data with histological examination to give an integrated diagnosis. This approach improves accuracy, especially for diffuse gliomas like astrocytomas, which can look similar under the microscope but differ greatly at the molecular level. Clinically, molecular profiling helps doctors classify tumors more precisely, which influences prognosis and treatment options. For example, astrocytomas with IDH mutations tend to have a better prognosis than those without. Certain molecular features can also predict how well a tumor might respond to specific therapies. For instance, if the tumor has methylation of the MAGMT promoter, it may respond better to chemotherapy drugs like temozolomide. Additionally, identifying specific genetic alterations allows patients to be enrolled in targeted clinical trials, paving the way for personalized medicine. Research continues into less invasive methods, such as blood tests that analyze tumor DNA or proteins circulating in the blood. While these are still experimental, they hold promise for future non-invasive diagnosis and monitoring of astrocytomas. In summary, molecular profiling for astrocytoma diagnosis is a sophisticated process that looks at the genetic and molecular features of tumor cells. It enhances traditional diagnosis, helps predict how the tumor will behave, and guides personalized treatment strategies. This approach is a vital part of modern brain tumor care, aiming to improve outcomes and support brain health. If you or someone you know is facing an astrocytoma diagnosis, it's important to consult with specialized healthcare professionals who can interpret these molecular tests and develop the best possible treatment plan based on the latest scientific evidence.